Hello, everyone. We're going to be starting shortly with this webinar on extracting gene disease evidence from literature, genetics, genomics, and more. Today with you is me, Maria, from Europe PMC, and Denise from Open Targets. Denise, you can say hello.
Hello.
So let us just very briefly outline some of the technical details. Some of them you can see on the slide, but if you would like to download the presentation, it's available under Materials section. All of you are currently muted, but if you would like to ask questions, you can post them in the group chat. Look out for the chat box and sorry, just a second, we will. All right, now can you see the slides? So you can post the questions in the chat box and we will try to answer them at the end of the webinar, but you can post a question anytime you like. And of course, very important to us is to hear about your thoughts on the webinar. So if you have any comments at the end, please fill out evaluation survey and we would really appreciate your feedback. So starting with the webinar, the reason we're talking to you today is that no matter what field you are currently working in, every modern day scientist predominantly communicate their findings in research publications. And if you want to find data that could help you to identify and prioritize new therapeutic targets, often you will have to search for evidence in published work. But with growing amount of literature available for analysis, it becomes increasingly difficult to extract useful information quickly and efficiently. On top of that, disease biology is a complex topic. It concerns physiological changes that are governed by a combination of genetic and environmental factors. If you wanted to get a comprehensive overview of various components involved in development of a disease, you might want to look at diverse biological data. Your workflow may require comparing sequence variants from healthy and diseased individuals, while also looking at the protein structure to see how mutations would influence protein folding, checking with a small molecule database to see if ligand binding would be disrupted, and perhaps scrolling through a review describing the disease symptoms. It becomes harder to integrate these diverse data formats into a coherent workflow. We at Open Targets and Europe PMC want to help scientists, particularly those working in the drug discovery field, to keep up with emerging evidence on gene disease associations and provide tools that help integrate this dispersed evidence available via public datasets to help generate new therapeutic hypotheses. So what are Open Targets and Europe PMC? And I'll hand over to Denise.
Okay, so hello everyone. Yeah, thank you very much for joining us. So as Maria said, I'm Denise, and so I work with the Open Targets team. So I'm not sure how many of you perhaps have heard of Open Targets or seen anything about us, perhaps on social media. So Open Targets is actually a consortium between 6 different institutions or pharmaceutical companies. So Open Targets was launched in 2014, so it's quite a recent consortium, and it was founded originally by GSK, EMBL-EBI, where both Maria and I are talking from, and also the Sanger Institute. 2 years later, then we had Biogen, which is a biotech company in the US. So they joined the partnership. And more recently, in 2017, we had Takeda on board. And just 2 months ago, or perhaps even less than that, we had Celgene also joining the partnership. So all of those partners, so we've got around, perhaps 500 people working for Open Targets. We all share this vision to transform the way that drug discovery is carried out. So we want to make drug discovery a less painful process, and we want to do so by the systematic identification and selection of targets. And there are two components within Open Targets. There is the data generation component and also the data integration. And I'm going to focus on the data integration today. So one of the resources within Open Targets that is quite popular is the Open Targets platform. So it is the first resource that we have launched, which is, you can think of it as a one-stop shop. It's one place only that you go to, and you can get all sorts of different types of data integrated in that platform. So we've got genomics, genetics, transcriptomics, we've got text mining as well, drug information. So we have integrated all publicly available databases out there, integrated them into an easy-to-use web interface. And in addition to putting all those different data sources in one place, we have also added value to this integration by scoring target and disease associations, and also by providing links to the original sources. So the platform itself was launched in 2015, so just a year later than the consortium as a whole. We do have different releases every 2 months or so. And at the moment, so the latest release as of April 2018, so we've got information about 21 targets, almost 10,000 diseases. We made the associations between those two, and we've got more than 2 million associations, and the associations were made based on data. So the associations are data-driven, and we rely on lots and lots, millions and millions of evidence in order to make the association between targets and diseases. So this is our data model. So we've got targets here, and we've got diseases there. And the main objective of the Open Targets platform is to make the association between those two entities. And because we want to do this following a data-driven model, so we rely on pieces of evidence. So at the moment, as of April 2018, those are the different sources that we heavily rely on in order to make the associations. Those resources are all publicly available, and we have a mix of data containing germline variants, somatic mutation, drug information, and so on and so forth, and including text mining coming from Europe PMC. So I really recommend you guys to have a look at this documentation page here to get all the details on the different sources. And with that, I will hand over back to Maria so that she can explain a little bit more about Europe PMC.
Thank you, Denise. So Europe PMC is actually an open biomedical research literature database. It has been established as a partner of PubMed Central, together with the PubMed Central USA, that you might know as one of the resources at the NCBI. So Europe PMC, as a partner of PubMed Central, shares full text content, but we also provide access to other kinds of records. So in Europe PMC, you can find over 33 million articles, majority of them coming from PubMed, but also other sources, for example, additional resources for plant biologists, and patents, clinical guidelines, PhD theses, and other types of related content. The mission of Europe PMC is to support text-based innovation and develop tools that improve information retrieval. Now, we consider literature as an important part of big data in biology. As I mentioned, in Europe PMC, we host over 33 million publications. That is 10 times the number of genomic variants that you can access in DBVAR database, or 240 times the number of protein structures available in Protein Data Bank Europe. Moreover, journal articles are the major dissemination route for new findings. They contain facts and data that can be extremely valuable for future studies. Scientists generally access this evidence simply by reading. However, as you know, with the current publication speed, research on diabetes, for example, will produce about 70,000 papers a year. To scan through all of them, you would need to browse more than 7 papers every hour of every day without any breaks. So, how can your PMC and open targets help you extract useful evidence on potential drug targets without having to go to different places and scan through multiple data sources? Let's imagine you work on polycythemia vera. It's a rare neoplasm. Your colleague has recommended you a recent article that you can see on the slide, which reports the effects of a drug treatment in patients with this disease and a particular allele of JAK2 kinase. Now, perhaps this is a 20th article on your reading list for today, so you've got to spend your time wisely. What you really want to know is A, what evidence is presented in the paper, and B, what other evidence is available for implication of JAK2 in this particular cancer type. We'll start with the first question. Extracting evidence from publications is a difficult task, and text mining is often used to automate knowledge extraction. However, often the application of text mining is limited because of the technical know-how to set up a text mining pipeline. Not everybody has expertise to just do it in their own lab. So at Europe PMC, we wanted to help bioinformaticians and web lab researchers to overcome this problem. And our solution was a community text mining platform. Text analysis is done by experts in the field who share their results on the platform, where these results can be openly used by others. But how does that apply to disease biology and finding associations between targets and diseases? Well, text mining is like a sophisticated search. It allows you to find certain facts in the text. One simple example where text mining can be useful is locating biological entities in the paper. These entities can include proteins or chemicals or even processes like cell division. Concepts found in the text are tagged or annotated. In a way, a text mining annotation is simply a label that tells us that in this particular sentence that you see on the slide, p53 is a protein and methotrexate is a chemical. But this is perhaps the most simplistic approach to text mining. One can do much more complicated things with it. And for example, you can look at relationships between concepts. In this particular case, methotrexate acts as an inhibitor of p53, and you could capture that information as well. For the purposes of today's webinar, we will look closely at one of the text-mined annotation types in Europe PMC, namely the gene-disease associations. So we would like to know what evidence is presented in the paper on JAK2 allele. We will search for that paper in Europe PMC, and I'm going to now show you how it's done directly on the website. So we're going to have a small demo. So here I have Europe PMC's website. We can search for the particular paper that we're interested in. Now, just to make it easier, I have already searched for the paper with the title here in the search bar, and you can see that the first result is the paper that we've looked at before. And we will go directly to the publication. So here is a paper that you might want to read, and here is the abstract. What I want to draw your attention to is the menu on the right-hand side here that says Show Annotations in this abstract. What will happen as you click on any of these instances, and you can see here there are chemicals, diseases, gene disease associations, and other types of annotations found in this article, is that those will be found in the article text. So gene disease associations that have been text mined include an instance with JAK2 being implicated in polycythemia vera and another instance of the same type of association. Now, if you go to the full text of the article, you can see that there are perhaps more instances, so 4 found here, and you can navigate between those in the text. So there are other types of annotations that you can see here on the slide, and I'll just briefly show you the full list of them. So annotation types include genes and proteins, chemicals, organisms, diseases, gene ontology terms, accession numbers, genetic mutations, gene-disease associations, gene functions, protein-protein interactions, transcription factor target gene associations, and phosphorylation events. Now, all these can be found in articles in a similar way that you have just seen in the demo. So I'll go back here. So what it allows you to do is quickly scan an article for concepts that are associated with your topic of interest and assess if there is any additional useful evidence in this article. Now, what will happen if you click on the annotation is that you can see further information available from the Open Targets platform for this particular association. And so we'll go to the disease description available on Open Targets, and I'm going to hand over to Denise for this.
Okay, thank you, Maria. So let's just wait for the page to load. Shouldn't take too long. So here we are. So yeah, so if you're coming from the Europe PMC website, so you can get a link to the page in Open Targets. So this is what in Open Targets is called as the associations page. So what we see here is a list of all the targets associated with my disease of interest. So the association is done based on data, so it's a data-driven model. We've got the different types of data are listed here on the left-hand side. You can do lots of different things on this page. So within genetic associations, we have 2 types of germline variants. We've got mutations coming from 3 different data sources. Remember, this is a type of cancer, so you will have some somatic mutations implicated in cancer that can be used for the association between several genes and a given disease. We've got some drug information as well, so you can find out which drugs from the Campbell database are already marketed or approved, at least for some clinical trials. We've got animal models and so on and so forth. So as it stands from the April release of Open Targets, we've got just under 500 targets associated with this disease, and including, so you can scroll down here. So we show the first 50 targets for this disease, and of course you can always explore further targets down the table there, and we rank the targets. So out of those almost 500, we rank them according to the association score. But what you can do here, you can also filter, in addition to filtering by the different data types, you can filter by the pathways that the target is involved with. You can also filter perhaps on the class of the target. So perhaps you are interested in targets that are a membrane receptor, or perhaps they are an ion channel. So we give that flexibility for you. You can also filter the targets depending on the tissue that the target is expressed. So there are different options here that you can explore. And you can also, if you are working on a given disease, you can also input a list of targets that you know already are implicated with that disease, and you can find the information that Open Targets has integrated for you. So in this, if you go back, if you remember the use case scenario that we started off with, so we had this user who was presented with this paper that showed an association between polycythemia vera and JAK2. So JAK2, according to the Open Targets platform, is actually the top hit for the association. So it's a score of 1. We've got information at the genetic level. We've got drug information. We don't have anything in terms of pathways affected by mutations or data on differential RNA expression, and we do have text mining data coming from Europe PMC. So you can click on any of those rows and you can explore the evidence behind the association. So there are a few things that you can do here. If you want to jump straight to the text mining, you just click on that row there. This will take you to what we call the evidence page. So it's listing all the evidence, all the data that Open Targets can get hold of, and that can be used for the association between JAK2 and polycythemia vera. So we've got the papers there coming from Europe PMC. You can go back and explore additional papers. They can be sorted by the year of publication. You can also download this information as a table, separated value. You can then collapse this tab and perhaps explore other types of information there. And this flower is trying to represent the data available for the association. So in addition to text mining, we also have those 3 types of data. And then from here, you can explore a bit further. There's plenty of things that you can do. It's a very intuitive and easy to use interface. So you can see all the variants that map to JAK2 and perhaps are associated with other diseases. You can also see the variants here highlighted as a lollipop. When you have variants or lollipops in red, they're actually variants that map to that region of the genome, and they are associated with my disease of interest here. We also have somatic mutations, so again, feel free to explore this once the webinar is done, and we always link back to the provenance of the data, so the original source of the data. So we integrate the data, we show the data integrated in a easy to use interface, we rank the data, we add value by providing an association score, but we always link back to the provenance of the data so that you can make your own judgmental call. We also, in terms of drugs, yes, as I said, all the drugs come from Campbell. They are in different clinical phases of trials, some of them already marketed. And from here, you can also explore more. For example, you can get information at the drug level as well. So we've got around 18 different data sources within the Open Targets platform, and they are all publicly available. Similar data sources are combined, are grouped together into major data types, as we call them. So we've got similar data sources coming from Uniprot, EVA, which is similar to DBSNP, Genomics England, GWAS Catalog, PHIWAS, and so on and so forth. So all those sources, they give one type of information with germline variants. So they are combined into this larger umbrella of genetic associations. And the same happens for mutations implicated in cancer. So they come from different sources, but they are combined and grouped together as under somatic mutations. So those are the different data types you can see on the platform, and you can also filter by the individual data sources. So we give you the options there to choose from. So by no means text mining coming from Europe PMC is the only data that we use for our associations. So I've been talking a lot about targets, about diseases, and about data being used for the association of them. So some of you perhaps may be wondering what Open Targets consider as targets. So what is the definition for Open Targets of a target? So a target, it can be a non-coding gene. So it can be RNA genes, microRNA genes. It can also be a protein coding gene. So you can search for ensemble gene IDs. You can also search for uniprot IDs. You can search for the human official gene nomenclature for genes, such as, for example, DMD. So those are the entities that in open targets we always refer to as targets. So they can be coding or not, and they can be searched by any of those different IDs. And you can also search for synonyms as well. So those are the targets out of the way. Now, when it comes to diseases, so you may remember from one of my first slides, I did say that we've got just under 10,000 diseases in the April release of the Open Targets platform. So the diseases, in order for us to integrate the data, we need the diseases to be annotated and classified following an ontology. And the ontology that Open Targets has chosen is the EFO. So EFO stands for Experimental Factor Ontology. So an ontology allows us to classify terms. In this case, it allows us to classify diseases, and it provides a controlled vocabulary, and it also provides the relationships between diseases. So in this example here, I'm showing you asthma, highlighted here in green. So there can be several types of asthma, such as childhood onset asthma. So this within an ontology is known as a child term of asthma. So asthma is a disease of the respiratory system. So that's why we can, by relying on ontology, we can take advantage of this hierarchy, and we can show our users the relationship between all those different layers, including the children terms and the parental terms of a given disease. So the advantage of using an ontology is that it promotes consistency. It also allows for an easier and automatic integration, because as Maria said, there is such a deluge of information out there, so we cannot do this manually. So we need to automate this somehow. And by relying on ontology terms, terms such as this that you can see on the right-hand side here, so the EFO term, it is consistent across the releases, it doesn't change, it's unique as well. So you can search for those terms, or you can search for the disease name, and we can provide consistency across different data sources and across different versions of our database. And it's also important for Open Targets to rely on an ontology, simply because sometimes there can be some piece of evidence available for a child term of a given disease. So by relying on that piece of evidence, we can, as we call it, we can propagate the evidence from lower terms within the ontology graph to higher terms within that ontology. So you can think of it as indirect and direct evidence. So Open Targets does rely on both types of evidence, those that are reported directly, but those that are propagated from lower terms within the ontology. You also may remember that I've been saying that Open Targets does add value. So it's not a one-stop shop where you can get all sorts of different types of data sources or data types. So we also try to add value for our users. So it's all nice and perhaps straightforward to make an association between targets and diseases, but we want to allow our users to rank those associations, and also perhaps to focus on the associations that do matter. Users are indeed very concerned, and they want to know the relative weight of the evidence for those different targets associated with a given disease. So we do provide this association score, and as it stands, we go through a 4-tier system of scoring. First of all, we look at the evidence. So we look, for example, at a given SNP, one single SNP coming from a GWAS paper, and we score that evidence. So we look for how many cases have got that SNP that have been reported for. We also look at the p-value of that association, and we also look for what this SNP does at the genome level. So does the SNP cause a change at the amino acid level? Is it an intronic SNP? So all those pieces of information are recorded and used for the scoring at the evidence level. Then the second tier is when we combine all the different SNPs coming from one data source. So there are many papers out there reporting many different SNPs associated with one single trait. So we combine all those different SNPs for that single trait, and then we give you the association score at the data source level. So all SNPs coming from the GWAS catalog. However, as you may remember, we do combine different data sources. They are different but similar in a way because they provide a set of SNPs. So we will then combine all the scores coming from the GWAS catalog, coming from EVA, coming from PHIWAS, and all the other sources within genetic associations. So this is the third level of scoring. And then we combine everything together to give you what we call the overall association score. So by using this mathematical formula, we can also account for replication of data, redundancy of data as well. The scoring framework on the platforms, we do apply different weight to those different sources, and you can sort the sources according to decreasing or increasing level of the score, and you can perhaps just focus on genetic associations or text mining as you wish. So I think with this, I'll hand over back to Maria. She will then talk a little bit more about the text mining evidence.
Coming back to text mining, I just wanted to sort of dissuade you about the text mining being not a reliable data source, because this is a concern that some of the users have, and it is a valid concern. So I would like to just highlight how the text mining is done so that you know how we obtain this evidence and how certain we can be about it. So in this particular case, in order for us to say that there is a gene-disease association present in a paper, we set up the text mining algorithm to search for mentions of a gene and a disease in the same sentence. But of course, if we would just find all instances like that, that might contain a lot of false positives. So we do a number of filters for this. We only look at primary research articles. There is no reviews or commentaries or letters to the editor included in this. We only look at introduction, results, and discussion. No materials and methods, no references, no funding statements, nothing like that. Associations have to appear more than once in a paper, unless they're mentioned in a title or abstract, which we consider as sort of more important parts of the paper. As Denise mentioned, we do use defined vocabularies for genes and disease names so that we can harmonize the mentions. We filter out entries that can be too ambiguous. For example, if a gene name is A, or it's a protein name large, we cannot be sure that in a sentence it's actually a protein that is mentioned, so we ignore those, and we include some term variations. And there is a number of other rules, and if you're really interested in how this works, I've included a reference to the article which describes this at the bottom of the slide. But basically, for each association, we then calculate a confidence score. And it takes into account where the association is found in the paper and how many times we could find it. And I can tell you that at least 50 to 80% of other evidence overlaps with the literature mining data, which means that we can be confident about the annotations that we provide, but also that there is part of the data that it cannot be found using other types of evidence. It's only present in the literature. Now, this is, of course, up to you to explore it further, but that's maybe where new and interesting targets may lay. And so I just wanted to mention briefly that, of course, although we set up stringent checks for text mining, sometimes it can be difficult, especially if you're looking at abbreviations. And so it can happen that you notice an association that does not look right. So I'll just quickly show you how you can actually report such an association. So here I have a paper, and if you look at gene disease open targets, as I've done previously, and you highlight it, and you look at the sentence itself, it says inflammation, cytotoxicity, and anticholinesterase assays. So what the text mining platform has done is it linked cholinesterase with inflammation. But of course, that is an incorrect annotation, and you can report that. So this is like a crowdsourcing platform, because you can improve the quality of annotations, and so you can report a problem with one or endorse a useful annotation, so that the text mining will be improved in general, and further annotations will be of higher quality than the previous ones. Another thing that I would like to mention is that, unlike many other providers that only text mine abstracts, we actually mine the full text of the articles to search for associations. So we may find more associations for you, but there is an issue with article license, meaning who owns the copyright to the paper. So for some papers, you will not be able to see the associations. Just mentioning it, that you're not surprised if you don't find them. So I'll hand over to Denise, and we will actually do a couple of use cases, starting from disease and starting from publication.
Okay, so let's suppose, so you started off with that single paper, and then so you're interested in polycythemia vera, and then so you can come to the Open Targets platform and simply search for that. So you can find all the other targets associated with that disease, and you can find the evidence for the association as well. So I think actually we have already covered most of this, but yeah, so I would just, yeah, so this is my disease of interest. And as I said, you can explore this. You can also view the profile page of the disease. You can see whether or not there are similar diseases to the one that I'm interested in. And this is based on the set of common targets, or the set of targets that are common between the two diseases. Then you can explore a lot more here, so you can see the exact drug information that is available for a given association. As I said, you can also jump to the drug page within Open Targets. So there is plenty here that you can do. And go back, you can also explore the target outside any context of a disease. So you can see every single drug that is available out there to modulate this target. So if you scroll down here, you can also filter. So perhaps you are interested in one type of sarcoma there. So you can see what is the drug. You can remove that filter. You can perhaps just focus on clinical trials that are currently on phase 2. We've got protein information as well coming from UniProt, so it's a quite nice diagram of the protein as well, so you can see exactly where the transmembrane domain lies. So there is plenty here that you can explore. So basically within the Open Targets platform, you can start off either with a disease, or you can start off from the gene or the target point of view. So if you start from the target point of view, you will see 3 different views. So we've got the bubbles view, so you can again click on any of those small bubbles, get more information on the evidence for the association. You can perhaps choose to see this information as a table. You can download the data if you want to, and we've got also this tree view. So I think perhaps, yeah, Maria will carry on with her next, and I think the last demo.
Yes, so thank you, Denise. And I come back to starting from a publication. Now, I know I've shown you before the way you can highlight gene disease associations in a paper, and that can be useful for scanning, but that's not where it gives you most functionality. What you actually want is information retrieval. You want to be able to search for specific publications that might be of interest to you, and you can tailor it very precisely using these text mining tools. So you can answer questions like which studies report gene mutations implicated in diabetes, or which articles on P53 cite clinical trials. Perhaps the most useful way to explore it is actually using the programmatic access. So Annotations API gives you an opportunity to explore all the annotations that I have mentioned, gene disease associations, but also others, protein interactions or model organisms, and put them together in a coherent workflow. If you don't have programmatic skills, don't worry, I'll cover a bit on what you can do on the website. But if you would like to learn how to do this, you can go to europemc.org/annotations API, and you can explore it using annotations by relationship. So this is where you can search, for example, for papers where P53 is connected to cancer, and then search for what papers or what other annotations can be found there. So this is for the more programmatically adept people among you, and I can share a link to a webinar that explored this in more detail. But coming back to the website and actually trying out what you can do as a wet lab researcher in your everyday information search. So if you go to advanced search in your PMC, what you can do is find a section called external links. And under external links, select Open Targets platform. Now, if we search for this, it will bring you all papers that have reported a gene disease association. So if you add this syntax to your everyday search for papers, you will pull out publications that specifically have been text mined and found that they contain this particular type of evidence. So you can then, for example, combine it with diabetes to search for papers that provide associations on diabetes. You can sort papers by date or time cited to pull out perhaps more influential publications. Another thing that can be quite useful is combining this type of search with search for publications that report other types of evidence. Now, just very briefly coming back to the publication, in your PMC, it is possible to pick up data citations. And data citations that we find are found via accession numbers. These are unique IDs, or if you wish, data tags that are assigned to data set in a repository. If somebody has sequenced a genome of a new model organism and has put that sequence in a database, that sequence gets a unique ID, an accession number, and we search for these accession numbers. So this is what you can do in your PMC as well. You can go back to the advanced search and select data links and data citations. And here you can see, for example, clinical trial citations, or you can select protein structure citations or reference sequence variations, any kind of data that you might be interested in. There's also a useful trick to search for publications that mention something in a specific section. For example, if you want to see papers that report results on a specific mutation, you can select the section type and here pick results. And then we say papers that mention diabetes in the results section. So there's a lot of different ways in which you can combine these things in your PMC. And I'm gonna just leave it at that, but I'll post a couple of links to other webinars that you can explore this in more detail. So now we're at the end of the webinar, and I'll hand over to Denise to just briefly conclude.
Yeah, so I think we are running a little bit out of time, so we better just try to conclude the webinar and perhaps share you with the take-home message of this webinar. So I think we both, Maria and I, we believe that we are all very much baffled by the deluge of information that is out there in science, whether you work in drug discovery or anything else really. So I think the take-home message is don't be. There is hope and there is help, especially with Europe PMC and also open targets.
Thank you all so much for joining us today. We hope it has been helpful to you. Take care, all the best, and enjoy.
Thank you. Bye.